moving on. So have we? There we go. Starting again. So the technology here, we have unlimited logons. If you have your own computer, it's very easy to log on. I'll send you the link to the software, particularly if there's an assignment to do this. Today, this is just introductory stuff. This is called the iWork system. You remember we did it a little bit with when Dr. Buxton was there. Today, and just to show it to you, we'll be doing this. This is what they call a foundations lab, not unlike what we did with him. And we're going to be, and we just have one thing, as you can see, to plug in here. There's nothing major involved with it that's there. It's this thing. And basically, we're going to do the reaction time to sight and sound. And so, in your mind, the question is, do you react more rapidly to a visual prompt or to an auditory prompt? That's really what this is about. And so you think, and it's almost, some students get it right, some students get it right. There's no right or wrong, by the way. It's just, that's really what we'll be utilizing today. Most of the time they give you this, so I need one subject and one person to basically run the test. The subject will be looking at the screen or just listening. And they'll be looking at the screen for a line, we're listening. And so and we'll get to that when we find it. So I'll send you, we have unlimited logons. We all use the same logon that I created 7 years ago. So we've been using this off and on for 7 years. The difficulty, that was 2019, and it worked pretty well. And then all of a sudden in came COVID. And it was really kind of difficult because we were off site a lot of the time. But, there's a lot. I'll be doing electrocardiograms with you. I'll be doing pulmonary function studies, some nerve-related studies. We'll do nothing invasive about this. We're not going to shock you. We're not going to stick you with a needle, despite Dr. Badger. See, Dr. Dress, when he would have let me do this. But Dr. Badger, he's a stickler. He's fond of you guys. You don't believe me. So you always get this. So, for instance, now I'm just popping up that one. But when you look at, this is the iWorks, the criteria that is, and you'll see what'll happen is I'll start this, and it goes at a particular time, and I'll be, utilizing, let's say, something that will generate a mark when I read the protocol, and someone will have to respond like this, and you can see that like that. And so you'll either be hearing a prompt or you'll be seeing a prompt visually, and we'll compare how fast. And this gives you, as you can see, it's calibrating below are you stopped now? Stop. So you can begin to see this, and we can calculate how it is from, what the amount of time that was there that was involved. Probably today I'm not. Oh, it's okay. It'll be fine. So that's kind of what we're doing with regard to this whole issue. And so I'll send you the information. So if you're going to do one, if you don't want to put it on you can load it and unload it. It's just, you have to load a file, execute the file, sign in. It's got a fairly easy login. I think it uses my email, and you'll see the password that's associated with it. So you can put it on there, and you can analyze it yourselves if you want to, or do so in a group. Easy enough to do. And if you're having some difficulty with it, I'll show you, and I can work. The tech people are supposed to have it on, but I'll remind them, on university computers. So if in the library or in a computer lab, they would have this already installed. So you can just click on it the way I have it on my, wherever it is here, LabScribe. What it is, and probably said LabScribe 24 now. And you'll click on that, and it will take you, and then you just have to put your login credentials. Don't create your own. They yelled at me already about that. Oy. It's one of those. But that was years ago, so I haven't had the problem since. Good. So that's everything really you have to know. As usual, the exam, I'll announce them. Uh, Most like, if you're taking it in class and it's closed book, I will certainly provide one of those study guides and things like that. The usuals. I'm not changing. Questions? No? Good. Thank you. I don't need the distractions today. Let me get on with it. So for those that didn't have anatomy, this is the eye anatomy part of the program, and we're going to delve into eye physiology. It's there. So this is the chapter on peripheral nervous system. So you can see what's going on. On mine, when you look on the computer, it's a little bit different. And this is just some of the same stuff that we had gone over previously. But I want to get to the eye without going over all of these things and the different kinds of receptors that are there. So spare me because I want to do the eye part first. And so this is all the eye anatomy. Everybody remembers that we dissected the eye. For the four of you that weren't here to dissect the eye, if you need clarification, just ask me or ask somebody who did it. So you understand the structures we're talking about when we get there. Effectively, we're going to delve inside the eye, not dealing with the anatomy. We're getting to the point now, and the same thing, and all of this is anatomical. We want to get into the physiology. We're not going to talk about the extraocular muscles or anything like that. There's certainly abnormalities there would play a significant role with regard to this. And so now we're getting sort of where we want to go. And so if you're looking up here, you remember this, even though it's anatomical, we want understand that when the light comes in, depending on where it contacts, and remember that fovea centralis and the macula are the areas where if you have corrective lenses or if you have good vision, where it goes, that's your best visual impulses. And understand remember, that's the retina, that relatively thin, almost delicate layer that we saw in the sheep or the cow's eye, which we did. It goes through that layer, and you're going to see it's almost like it's in those. We're going to see the rods and the cones are almost embedded into that pigmented layer. And it's really designed to avoid scatter. And again, I'm not going to go crazy about focusing it and the lens because I dealt with that more in anatomy that are there. So I thought you should be familiar with that. If there's anything on topics that are anatomical, I will certainly let you know. So we're really dealing with the retina, and we're dealing with the photoreceptor cells that are there. And again, this is when it's very delicate. That signal is going to go through the retina and go to this outer pigmented layer. That means the light doesn't hit immediately. It goes through to that outer pigmented layer, and then that's where the signal gets triggered. So, and we'll get to that just ever so shortly. So this, as you can see, a single cell thick lining next to the choroid, that vascular layer that's there, and extends anteriorly all the way to that ciliary body. You might recall that is here, the extension. You can see where the retina, they're trying to give it to you diagrammatically, almost to that area. Remember that serrated edge? Okay, that was there. So it extends rather generously anteriorly in that projection. So all the way, it covers that ciliary body, all the way to the iris. Remember, that's the pigmented area of your eye. It's there, absorbs the light, prevents scattering. It gobbles up the when photoreceptors are triggered, they fragment. So when they're triggered, basically we're constantly rebuilding. We're breaking them down and rebuilding them all the time. All the time. Vitamin A is stored there, but basically you're going to see this consistently. It's magic. When it was always magic because someone would be exposed to a big flash. All of a sudden, in the old days, we'd be starting a gas heater or something like that with a match or like a long match. And all of a sudden, and there'd be almost like an explosion, the light. And the treatment is not unlike, you basically take away everything. You make sure there's no light for about 48 hours that they can get to, none whatsoever, and allow the eye to heal itself by gobbling up all the fragments and rebuilding new rods and cones. Yes, ma'am.
Is that the same for like when you're welding and stuff?
Yes.
You have to have a lot of it.
Yeah. So much protection for that. Okay. I mean, that's what you got to hold it. I know a little bit about that, that's there, but you're just that constantly, and not, and also with anything that's coming your way, you mean little sparks and things that could cause burns on the surface would be problematic at best. I know this son was actually looking at, was taking welding at CCAC for a while. He didn't end up going to that field, but I mean, I know there was a lot of.
I was doing it too, but I didn't end up going.
Yeah, he was going to do that underwater welding type stuff. I'm kind of interested in that. So moving on, that's part of it. So the neural layer, very transparent, runs that ora serrata. Then we get to different receptors. The photoreceptor anatomically, the bipolar cells we talked about earlier today, and the ganglionic cells that were there. And it spreads from those photoreceptors from peripheral to central, from outside to in, as you'll see. And the ganglionic cells, they're long, just like we talked about, those long tails, those transmitting tails of axons. Those axons eventually become what you and I call the optic nerve. And the optic nerve is transmitting it to the thalamus, and the thalamus sends it by what's called the optic radiation to the occipital lobe for processing. So we start to see things there, and you'll see the anatomy of it. And I'll go into it. I apologize again. I'm so distracted, but we'll get there. So, and again, the optic disc or blind spot, and that's really where they're bringing all the nerves to. And we look at the rods and the cones. There are 20 more times cones than rods. Beg your pardon, the other way around. 20 more times rods than cones. The cones require a lot of light. The rods are what give us night vision. The rods we're using when there's limited light that's there. It's almost like they're black and white grayscale. And the cones, CO for cone, CO for color. A band, I band. It's not hard. So the rods, dim light, more numerous by a considerable amount, not particularly sharp. Again, I think I told you that. It's like how many times, it happened the other day. My wife and I are driving, and all of a sudden I catch out of, when there's fairly light, and you see a deer feeding on the side of a hill, falling. It's like, oh, because you caught it out of your peripheral vision. Those are rods. You don't need a lot of light. Cones, different, macula lutea, that area near the blind spot and the deep little pit in it, that's your highest vision. And so this is the look-see. So you have, and you can see the way they've done it diagrammatically, cones because of their conical shape. Their pointed shape, and the rods more of the conical shape, but without that sharp pointed end. Here you can see that pigmented layer of the retina. And then as we move along, now you can see the abundance of rods compared to the cones. So they're going to alter. The photopigments are going to change. When they do so, it's going to generate an action potential. Interestingly, it's not a depolarized. Mostly we think when nerve fires, it's depolarized, closer to threshold. It's almost like these are always firing, and when we block it, it actually creates a signal. So we're causing hyperpolarization. Very unique. There's not a lot of systems that do that the signal comes to you because something stopped transmitting. It's almost like I don't hear that buzz anymore. Whoever looks something's wrong. That. The bipolar cells basically signal transduction, taking whatever that signal is, making it into a different form that can be interpreted visually. And here are the ganglionic cells. The cell bodies are there. And this long ganglion heading back, effectively an axon, and multiple ones, eventually all forming what we call the optic nerve. And so you can see it visually this way with the pigment. You can see the nuclei of the rods and cones, the bipolar cells, nuclei, and the axons of the ganglionic cells, all there. So it's pretty easily understood in that regard. So the blood supply comes from the outer third, photoreceptors, and as you see, and if you look schematically and go back and look at the optic nerve, nerves have the blood supply in the middle. So the blood supply, and that's what's supplying the inner two-thirds, more or less. Okay. I commented on those before. Those are the only place where you can actually, with now we do it with technology, but within old days, with an ophthalmoscope, where you could see the interior of the the interior of arterioles visibly. So arterioles are hard; they're so tiny, but here you can see them. And I said this before: what's happening in one arteriole is happening all over. We have a lot of different names. They call it the fundoscopic exam. This is an ophthalmoscope. Now they have technology; they can do it without having to dilate your eye, which they used to have to do. But basically, you don't want to see irregularities. You don't want to see sharp edges. You don't want to see little fragmentation, because that would suggest that those arterioles are damaged. So one's in your kidney are damaged, one's in your heart are damaged, one's going to your extremities are damaged. All of those, like in a diabetic, could be problematic. And a smoker. And for you folks who weren't here, and you guys might not remember it, as a heavy smoker, I had for an insurance physical in my mid-30s. My doctor's looking and he goes, my GP, my internist. He goes, Lou, I'm starting to see some change in those eyes I don't like. He said, you really have to think about stopping smoking. It only took me another 20 years, but that's not relevant. And then when I had a recent eye exam within the last 8 years, and I was amazed when the optometrist looked at me and goes, it looks like you were a smoker, but it's not. I can see improvement. And I had stopped smoking probably for 10 years at that time. So I stopped in 2008. That's probably 2018. Wow. So there's, if you catch it before it's too bad, because for instance, diabetics, the leading cause of blindness, diabetes, leading cause of kidney disease, people on kidney transplant, diabetes. There's a recurrent theme here that we're going to get to. With diabetes, retinal detachment is a little bit different. It's mostly trauma than anything else. Things like that. So there's all sorts of different. I'm not that familiar with the treatments. In the spirit of transparency, the eye is not my long suit. Okay. I always will tell you, like we get to some things in repro and things like that. I do not have as much expertise as we would have when we talk about endocrine, blood, diabetes, heart. I know that blood vessels. I know stuff pretty well. Digestive and urinary. So again, we're not talking about the segments with the different humors that are there. Again, that was more covered with glaucoma. We did that more in anatomy than we're doing in physiology. And the same thing with the lens and the different fibers. So that's done. And the next escape is here. Oops, sorry. B is the rest of the eye. C is special, is taste and smell, which are very much related. And then lastly, and I did a little bit of that in anatomy. And then lastly, the ear. And I'm not going to have enough time today to get into that mechanism, I'm sure. Make it look good here. I don't do physics. Any questions about that? But effectively, understand we have green, red, and blue, different kinds of cones. And the rods, as you can see, fall somewhere in the middle. And when you look at the electromagnetic spectrum, here, shorter wavelengths, we're heading toward ultraviolet, and then ionizing radiation, and I'm sure you had this, gamma rays, X-rays, then gamma rays. And on the flip side, this is green, we're hitting to red, and then you're looking at infrared, microwaves, I think, are in that ball game and things like that. And the rods are in the middle. So we only have those three colors, and everything that we see is blends of those colors, to my understanding. And really what color blindness is, to simplify it, as much as I know about it, is you're missing some of these, genetically speaking. So if someone's doing a report on that, it's neat because I always forget. So other than looking at the thing with the dots, whatever they're called. So, and again, the whole idea of corrective lenses, bending the light, and again, refraction. With the microscope, I'm not going to go there. And again, not my expertise. The light is bent 3 times. It goes through convexity in your cornea. It goes through another convexity in the start of the lens, and then goes through a concavity at the posterior portion of your lens. That's the way it is. So it's bent or refracted 3 different times. The whole ball game is to get it to the right place. And you see what some will make the light go. It's just the nature of it. And again, you can tell I don't want to get into it. So the light is refracted 3 times, cornea, lens, and leaving. The major refractory power is in the cornea, but that stays the same, assuming that you don't have to do something to your cornea. It doesn't really change it. And so again, when we start talking about near and far vision, I'm nearsighted. The whole idea of 20/20 is that 20 feet, so distance beyond which no change in lens shape is needed for focusing, okay, is where we get that 20/20 type of thing, 20 being the height of what we can see, I think. We have one of those charts in the anatomy lab. I don't know how to use it. Full disclosure, the ciliary muscles are relaxed. That lens is as flat as it's going. Lens is as flat as it's going to be. But some people are. I'm nearsighted, so I can't see things far away as effectively. And my wife and I got to change my glasses. I've had this prescription for 3 years. Hint: as you get older, it gets a little harder. Like you're all fuzzy. Even you're sitting up close. Now I can't read what's on your sweatshirt, for instance, or your hat. I still can't read what's on that logo on your hat. Terrible. But I'm always intrigued reading the little stickers and stuff you have on the front of your computers. Yes.
With your prescription, what is like the axis part?
I have no
Because
idea.
mine just recently changed.
No
I
idea.
don't know what, like, I have like a negative and a positive.
I don't know. Short answer. And I have an astigmatism, which is a curvature thing. I don't freaking know. I'm not, like I said, eyes. Ask me about a blood vessel. I got you covered. It's good to say I don't know. Fine lens, focusing. Yeah. See, this is me. Loss of accommodation over age 50. Changing lens shape to increase refraction. So my lens is going, I've had enough. Another 25 years on top of 50. I'm tired of doing this. Get stronger glasses. Don't do any of this. And remember, we talked about convergence, that whole accommodation, constricting the pupils, and you're looking, and again, that's that eye exam thing where it goes, and your pupils are going to converge on that, and the pupils are going to get smaller, and your eyes are going to rotate medially. So, I mean, it's just good to remember that. Not something I would ask you about. And we see things upside down and backwards. That whole letter E, did you do the letter E thing like in high school with the microscope and all that? Yeah. The little hairs and stuff. Oh, God. Help me. So myopia, nearsighted, hyperopia, farsighted, astigmatism. I always thought it was 2 words, like a lot of people did. Unequal curvatures. And so you can, and those all have to do with lens grinding, as far as I know. Those types of things. And as somebody's making your glasses, that's what I would do. They'll probably have somebody who's actually interested in this. And that's what, when you hear about LASIK procedure, they're basically making laser slashes. A laser is a very sophisticated knife, a cutting laser, that you can make very exact cuts. And they do that to try to change the curvature of your eyeball. For instance, and the beauty of it is you can, if it works, I mean, you don't have to have glasses at all. You can go back and buy whatever sunglasses you want. Rather than me, I get these ones when I go out, the transitions. So the photoreceptors themselves, and that's the point. I mean, that's the point I want to discuss here. Modified nerves that resemble upside down epithelial cells The tips are embedded in that outer pigmented layer. And so rather than going in the cell body and synaptic terminal just to see what it is, basically, whether it's a rod or a cone, it's layers of these photopigments that when they're exposed to light change shape, for lack of a better way to express it. So the photopigments change shape as they absorb light, and the inner segment is basically effectively is going to be the way it's going to conduct the signal, for a lack of a better way to express it. And they'll show it to you. So we're looking here, and that will change, and eventually it will deliver the signal to there. So I don't go crazy about the synaptic terminal, these inner fibers. The photopigments are in that series of discs that you can see, if you look close up on the rods, and they change shape. They degenerate fairly easily. The intense light, that's what I was talking about. I'm probably going to talk about the welding with that analogy. They're destroyed fairly easily, okay? And they're renewed, as you can see, vision is maintained because the outer segments are renewed. The tick fragments are gobbled up. So if you have a lot of damage, it takes X amount of time for those fragments before you start thinking somebody's permanently lost their vision, which is scary. And again, I could have worn contacts. My sister got contacts as soon as they were available. Okay, because we had really substantially eyeless in one eye, and I was afraid to put anything in my eye. Like my vision is part of what I am. I'm a very visually oriented person. Just me. So the notion of me sticking a contact, it's like, I couldn't do that. So anybody who's able to do that, good for you. It's interesting. She went to work. She spent the better part of 30 years working for the biggest contact lens people in the United States, Bausch and Lomb, up in Rochester, New York. So rods are very sensitive to light. That's why night vision and peripheral vision, they have a single pigment. So it's great. It's basically grayscale. The old days, like before we had color printers, you'd grayscale them. Okay, kind of fuzzy. 100 rods may converge into one ganglion. So it's not very, if you're going to see cones, very, very specific. So cones, low sensitivity, got to have a lot of light, react more quickly, 1 of 3 pigments, non-converging, very detailed. Some even have their own ganglion cell, so it's very, very accurate with regard to images. Imagine the detail we look at. Think of high def compared to the old days of TV. It's remarkable. I can go back to the days color TVs came out in maybe 63, 64. I started seeing this. So we're talking about 60. You have no idea, of what TVs looked like before. But black and white is very clever for movies. You're not looking at me like you agree with that. Oh, yeah, dude. It was like Spielberg doing Schindler's List, all in black and white, except the little girl in the red coat. Anybody see that movie? Oh, you're missing. Okay, not that long ago. So again, this is just summarize it. Color blindness, lack of 1 or more cone pigments, sex linked. We already aware with it. Much more common in males. A lot of males have them. So my dad was not unusually or so, and his vision was not. He wore glasses, but not sufficiently impaired to prevent things like military service or things like that. So, and he had the red green, as I remember, in which either the red or the green cones are absent, and etc. And you can see that it's just you have to rely on different shades to try to get some cues as to color. The key molecule is retinal, combines with 1 of 4 other proteins called opsins. They're from vitamin A. That's why vitamin A is so important. You see, eat a bunch of carrots because you have better vision. Because vitamin A carrots are a very good source of vitamin A. So, rhodopsin rods only. 4 opsins are in rhodopsin, so I remember that. 3 are found in cones, blue, green, blue, and red, depending on which wavelength they absorb. And so there's a certain amount, as you will see graphically, if you go back, they overlap. So, I mean, that's the beauty. We see so many remarkable variations in color these days. I mean, and again, when you see in these really good TV sets now, it's just and the computer, it's just extraordinary. So yellow light stimulates red and green cones, but if more reds are triggered, we see orange, for instance. So it's just like looking at a color wheel, if you want to think of it. Okay. And this is the difference. We have cis and trans forms. So the retinal isomers they're three-dimensionally different. Okay. Retinal is in a bent form in the dark, but when the pigment absorbs light, it straightens out. So it goes from cis to trans. I mean, I realize we use these terms now frequently in a different context, vis-a-vis gender identity and things like that, of which I know nothing about. Nothing. It's good to be old. So conversion of bent to straight, maybe that's relevant, initiates reactions that lead to electrical output. Try not to be offended by anything I say. It's all in you. And I said this and again, to speak of you folks who haven't been with me long. If I say anything that you're uncomfortable, please tell me. Everything I do is for humor or lack thereof. Just try to wake up over there, you two. And when it goes from bent to straight, it triggers the optic nerve. It's interesting. Pigments, process by which pigments capture photons of light energy converted into a graded receptor potential. What it basically means is, so that deep purple of rhodopsin in the rod, and it goes through, and all of them go through a similar process. After it's been triggered, okay, it's gobbled up, okay? So the pigment is made, it undergoes a process called bleaching, and then it regenerates. Similar in cones, but they're different types of opsins. I think the rod is the easiest to explain. So what happens, that particular opsin is in the cis form to form rhodopsin in dark. Okay, so first they make them. When it absorbs light, they call it bleaching, goes from cis to trans, and the retinal and the opsin separate, they start to break down. So, I mean, then they have to be gobbled up, recycled, and they regenerate. So all trans is eventually converted back to all cis, and the rhodopsin is regenerated in its outer segments. And they show you the pattern without going into the. Did you do any of this in biochem? This stuff? Anybody? Did they talk a little bit about Dr. Hilwig talk about this at all? So, I mean, you can see what happens now going from that bent form to the straight. So it just keeps going around continuously. That are there. And you can read that as well as I can read that to you, so I'm not going to try to do that. Pigment bleaching, regenerating, pigment synthesis, and we just keep doing it over and over again. So effectively, transduction is just energy transformations. We're taking it from light energy, and it happens to be a G protein pathway, okay, that's there. So, I mean, it activates it, cyclic GMP, et cetera, et cetera, et cetera. The big deal, and without going into all of the molecular biological aspects of it, suffice it to say, when that cyclic GMP breaks down, the channels close and the cell hyperpolarizes. So when we see something, the signal is disappearing, and that effectively triggers the next nerve in the sequence. Interestingly enough, it's not a depolarizing, but a hyperpolarizing signal. Exactly how that works, I can't explain to you as clearly as I would like to, but you can see what happens here as it goes along its merry pathway, typical G protein. The gated channel opens in the dark and it closes in the light, effectively. And so it's sort of, and because it's unusual, it's worth remembering. That's the kind of thing, whether it's on my exam or on an MCAT type of preparation test or some other kind of exam from a graduate perspective. Whenever you have oddities, they're usually good teaching points. Yeah. Things that are what we call counterintuitive. And so they're just showing it to you, what goes on with it, again, in detail. So we're going to trigger that thing, and it's hard to see this, and then go to the bipolar and then the ganglionic cell. And so you can just follow this down as much as you would like. And I can't really read it that terribly well. It shows my eyes. So the cyclic AMP gated channel opens, allowing the carbon influx to photoreceptor cell to polarizes. Then it triggers voltage gating. We're not going to have to know all this. Calcium channels here. The neurotransmitter is released, causes, as you can see, inhibitory postsynaptic potentials in the bipolar cell, causing it to hyperpolarize. Hyperpolarizing closes voltage gated channels, inhibiting neurotransmitter release. And so eventually we get none of these. And interestingly, it's sort of a reverse trigger. Go figure. You're confusing us. Sorry. So here, closed, so cation influx stops, the photoreceptor hyperpolarizes, and it changes everything. And say no neurotransmitter release. The lack of the inhibitory ones cause that to be polarized. The polarizing opens the voltage gated channels and generates the action potential. So one was before, one was after. Okay. If you want to look back, I'm not going to ask you about it because I don't understand it. If I have trouble explaining it to you, teaching it, I don't think it's appropriate for me to just say you should do that. It's just, you'll know. So I wanted to get through this. To me, it is comparatively, I understand the ear a little bit better, but not great. So it's very sensitive. The bleaching occurs even in starlight. In bright light, it goes so fast that the rods are virtually non-functional. So it's the cones that respond. So it's a matter, their rods are better with limited light. And so rods and cones depend on whether they're adapting to light or adapting to dark. Okay, when moving from darkness to light, you know what happens. We turn off the lights, and all of a sudden, we finish the presentation, and the lights go on, and you go, oh, right? Because of that strong stimulation, we visualize it a glare. I mean, we've all had that. That's what you typically, if you're going to turn the lights on, if you're doing like a dark presentation, you go, everybody, I'm going to flip the lights on. So you can close them until you kind of open them gradually. And obviously, the visual acuity returns to you. When moving from bright to dark, we see blackness because the cones don't work. The bright lights bleach rod pigments that are still turned off. Pupils dilate. The rhodopsin will get there, but it takes a fair amount of time. Everybody's different. Some people have really good night vision. I don't. I am. It's like my wife is like, pull over, but I'm driving. That's right. I'm chauffeured around. Everybody looks in the car and go, we understand. So that's me. I have a certain amount of that. Commonly caused by prolonged vitamin A, I don't know. Retinitis pigmentosa, you do not want to have. Degenerative retinal disease that destroys the rods or not replaced. It involves blindness. I do not know enough about it to talk about it. I can just anecdotally tell you this lady who was in the neighborhood where we lived and very, very close friends. Like when we had holiday dinners, she was always part of it. She's a widow and had suffered so bad. And I do not know if it's hereditary or not, but she ended up adopting a daughter. She was so concerned. And again, I would have to look that up and things like that. But she existed alone, but she knew everywhere in her home. She could cook. She was remarkable. But she was like part of the family. And so this, and anatomically, they trigger the optic nerve. Remember the optic chiasm? We saw that in the brain dissection. It's about 90-10, about 90% crossover, about 10% on the same side that are there. And so that gives us our depth perception. And so if we really, and again, I would do this more in advanced, in like, I did this in like graduate neuroanatomy, which I taught many, many years ago over at Chatham, where I was adjuncting and going through all of the different things that goes through the lateral geniculate and the optic radiation and the visual cord. We're not going to do it, but I'll show it to you. And all of these different nuclei that goes, so you can go crazy with this stuff. So basically, what you would, you could do a diagram. You can see that the outer portion, let's say, of this eye is here. And interestingly, most of it goes along the same side, where the medial portion crosses over. Same over here. Medial portion crosses over that lateral portion, same side. That's what gives us our sort of our three-dimensional vision. And when somebody loses an eye or has to wear a patch on their eye, they lose that depth perception. And what they'll do is they'll know where they'll intentionally put things in the same place. You go to a restaurant, you'll see they'll immediately feel where my knife and my fork. Things like that. And they can learn to drive. They just have to get an idea of when they get closer, see the signs, because of what they've done before. To remember what they say. There's a lot to it. When we used to do in advance, we put like here, if it was cut here, what part of the eye you couldn't see out of. And you could, and I would do those lab exams. I don't do them here because I think it's more advanced in nature. And you can see, and they diet here, optic chiasm, again, 90% or so crisscross, about 10% same side. And then they're into the thalamus. And then this is the radiation taking them back to the occipital lobe. So you can see anatomically how it was there. So depth perception in both eyes, this is what I was telling you about. So, etc. This is what I was telling you about this. Not getting into the different types. And the processing is very, very complex. To be able to identify, we first we're looking at when it's rods, it's basically size, shape, silhouette. Try to get an idea, and we can. Oh, that was a deer that's over there on the side. Because you have an eye, you can associate it, as opposed to the more elaborate information that we have. So we're processing it and generating this clear or somewhat clear picture, and then shuttling it forward for our frontal lobe to go, oh, that's not just deer, it's a doe, or that's a buck, or that's a fawn. Or it's coming across the street, stop. And so that really is sort of the summary of the eye. And we did this a little bit, and I'll go over it, but I think we'll do the experiment now, and we'll come back and I'll talk about the ear. So basically, the eye is a fairly complex mechanism. When we get to the ear, it has equal degree of complexity. This is sort of vis-a-vis the experiment. What it will be, so in the ear, remember, we're going to have the eardrum vibrates. There's going to 3 little bones in there. You remember from anatomy, the hammer, anvil, and stirrups, malleus, incus, and stapes. They're going to trigger something called the oval window. They're going to send that signal, and based on the frequency, we're going to hear loud or soft, but it's always going to trigger, and we're going to be able to basically transform it into words or sounds at the very least. And then if we are familiar with the sounds, it's like we're going to do with my great grandson. When they start to find, we're pointing at the dog, go dog, or our name for him, Doofus. Okay, so without further ado, we'll do this.